I guess we can start. It's a pleasure to introduce again Mike Lynch for his second lecture today. It will be about bioenergetics of cell parts. Please, Mike.
Okay, so I'll share screen. I'll follow up the end part of my last lecture, a little bit about the mitochondria, and then I'll go in and give a bit of a discussion. That's how we start to break down energy budgets of cells. So let me see if I can load up the first slide here. And I assume you can still see my pointer. Anybody see my pointer moving around? Oh, wait a minute. Let me put it into full screen. All right, well, I'll go ahead and get started. Last lecture, I discussed various ways that we go about trying to understand the total energy budget of cells. Showed you some examples of how the total cost of cells scales with cell volume. It seems to follow a general pattern across the tree of life, extending from prokaryotes to eukaryotes. But what differs is the rate of growth, which scales positively with cell size in bacteria and negatively with cell size in all eukaryotic groups that we've looked at. In general, the growth rates of eukaryotic cells, even in the same size range of bacteria, seems to be lower. So this is incompatible with the idea that somehow there was a bioenergetics revolution generated by the colonization of the primordial mitochondrion. And just to put things in contrast a bit here, a few people have made the argument that without the mitochondrion, there'd be no eukaryogenesis. So essentially every feature of eukarya owes its origin to the mitochondrion and its ability to generate more energy that would have been possible without it. So the basic idea here is, I think probably most of you know, the ultimate machine involved in making ATP is this crazy molecule called ATP synthase. It's effectively a turbine that sits in the membranes of cells. Protons you can think about this like a turbine in a dam, but instead of water going through the turbine to generate hydroelectric power, we've got protons going through. These protons are so this is a inner membrane and outer membrane of some cell. And we've got the electron transport chain here operating, and it results in the pumping out of protons into the lumen between the two membranes here. These then come back through ATP synthase, and there's this turbine here that whirls around, and every rotation gets you 3 ATPs. So in bacteria and in archaea, ATP synthase sits in the plasma membrane of the cell. So it sits out here in this little red ring. But when eukaryotes started to come around after the colonization of the mitochondrion, ATP synthase was its presence in the cell membrane was relinquished, and instead it became relocated into the mitochondrial inner membrane. And the idea here is that this might have been really good in terms of the overall bioenergetic capacity of a eukaryotic cell, because as cells get larger and larger and larger, you're reducing the amount of surface area per unit volume. You need that surface area for ATP synthase to be harbored. And one way around this might be to have all these internal membranes effectively in principle, dramatically increasing the surface area in which ATP synthase could colonize. And then that would enable us to produce a lot more ATP in larger cells. As I already pointed out, the growth rate data are inconsistent with eukaryotes having gained a major bioenergetics advantage. They're not growing faster than bacterial cells. And then as I'll show now, there's a diversity of other observations, including the anatomy of mitochondria, various aspects of ATP synthase and of ribosomes themselves that are inconsistent with the idea that the mitochondria is the use of mitochondria endows eukaryotes with some special bioenergetic capacity. Of course, it is the so-called powerhouse of the cell. This is the only way we can make well, it's the primary way we make ATP. So there's no question that it's an important part of eukaryotes, but it didn't necessarily launch us into a new bioenergetic world. One thing that you might not be aware of that I wanted to point out that's been discovered over the past few years. So just to back up, again, ATP synthase is this really complex molecule that sits in the inner membrane of plasma membrane or of a mitochondrial membrane. And there's this ring up here called the C ring. And it's through that ring that hydrogen ions pass through. And what's interesting is that the number, the C ring is what we would call it a homomeric ring. So there's multiple proteins that make these rings like this. So in pig and cow and sheep, in mammals, there's 8 subunits per ring. But there's other species ranging from various eukaryotes to some bacteria that have as many as 17 different proteins in these rings. This is an unsolved problem, but it's an interesting one because one complete rotation of a ring, the C ring here, in every case gets you 3 ATPs. So one way to think about this is that down here, you need 17 protons to make 3 ATPs. You can think about this as a very inefficient motor, a gas guzzler, if you like. Down here, I like to think of this as sort of the Prius of the ATP synthase world. Here, you only need 8 hydrogen ions to get your 3 ATPs. Why this kind of variation exists among species is completely unknown, and it doesn't appear to have any obvious phylogenetic relationship. The 8s here do happen to be all mammals, but if you go up and down this list, there's a plant here. These are both plants. All 3 of them are plants. There's various bacteria sprinkled throughout and so on. But what this tells us is that organisms, just based on the so-called proton motive force, have very different efficiencies of fuel utilization. Now, if the mitochondrion was going to save the day by providing more inner surface for the habitation of ATP synthase, then we ought to be able to go into eukaryotic cells, measure the total surface area of mitochondria, and compare that to the surface area of the cell. If the surface area of the mitochondria is no greater than the surface area of the cell, then it would appear that nothing has been gained by moving ATP synthase from the cell membrane into the organelles. And these are data from a wide variety of different species, unicellular species here in blue, and mammalian species here in red. And a one-to-one relationship would mean that the surface area of the mitochondria is no different than the surface area of the cell itself. And so you can see that virtually everybody is somewhere near that line. There's a couple exceptions, but they're only 5-fold more, and there are quite a few species below this. So there's no real gain in surface area in which to locate ATP synthase by using mitochondria. And this is really an exaggeration because mitochondria aren't sprinkled all over the inner surface area of cells. They're very punctate in their distribution. So there's these things called cristae that are invaginations that you can sort of see in these photomicrographs. And it's just at the tip of the cristae where the ATP synthases are harbored. So it doesn't look like geometrically there's much of a case for saying that that mitochondria provide more surface area for ATP synthase. We can look at this in a different way. Let me just move this top part over so I can see my own headings. One question we can ask is what do cells invest actually in mitochondria themselves? It costs a lot of energy to make mitochondria because they consist of a lot of double membranes. And here's a plot of the total membrane area of mitochondria versus cell volume, different kinds of cell sizes. And what we can see here is the total mitochondrial membrane area scales linear with cell volume across all eukaryotic species. So it's essentially isometric. You increase the volume of a cell and you increase proportionally the area of mitochondria. And my keyboard's frozen up again, so I'll have to get out and go back in. Is there a question?
Could you go over that argument about mitochondrial, the previous argument about mitochondrial area and cell plasma membrane area, and how it doesn't help to have many mitochondria for increasing surface?
Yeah, the basic idea is, if we go way back to this cartoon back here, the idea is if you're a bacterial cell, if you get larger and larger and larger, you've got more volume to support, but there's less surface area per unit volume. So the idea is you're running out of places to harbor ATP synthase if you want to maintain a very large cell. So the argument goes, one way to solve this problem would be to have internal membranes inside the cell. If you had many of these little bean-shaped mitochondria with all their inner surface area there, you're effectively increasing the area upon which ATP synthase can reside. You're making more plasma membrane, so to speak. Does that make sense?
You also said that this does not solve the problem. That's the part I didn't get.
Oh, yeah. So this argument sounds great. Makes sense geometrically, but when you actually look at the data in real organisms, what you find is the total surface area available for ATP synthase on mitochondria inside cells is no greater than the area of the plasma membrane itself. So you haven't really gained anything by moving your ATP synthase from the outer membrane of the cell to these inner membranes of mitochondria.
But presumably you've gained by not having to invest as much in biomass.
Well, maybe this will become a little clearer in a second, because the next point I'm going to make is that mitochondria themselves are very expensive because they consist of lots of lipids. So you still have to build these extra organelles here, and they're really quite expensive, as we'll see. So this gives us the scaling relationship. If you have a certain cell volume, this would be the surface area. This quantity here would be the surface area, follows this linear regression of mitochondria in those cells. And then if you know the surface area, you can calculate the total cost of membranes on mitochondria in the following way. This is getting a little ahead of ourselves. I'll talk about this when I switch to the next part of the lecture. But we know how many lipid molecules occupy a unit surface area. We know that because lipid molecules line up next to each other in parallel. And we know the area of what we call the head group. And so you can given the surface area, you can calculate the number of lipid molecules that must be in there. And of course, they're double membranes. I'll show you when I switch to the next lecture, we know what the biosynthetic cost of making a lipid molecule is. And I've just showed you the surface area and how that scales with cell volume. So the product of these gives, the cost here will be in terms of ATPs required to build membranes. The product of these is the total cost of membranes. I'm sort of jumping over a lot of technical details and not showing the actual numbers on each step here. But the bottom line is when you work this all up, you find that the relative to the total cell's energy budget, we have to scale the cost of anything in a cell relative to this total cell energy budget. The cost of mitochondrial membranes comes out to be this. So it's 0.05 times the volume to a very small number. So effectively, the volume is irrelevant here. And so this is equivalent to 5% of the cell's energy budget. So what this is telling us is the mitochondria themselves, just the production of the lipid membranes in mitochondria is 5% of the energy budget of a typical eukaryotic cell. So this is a pretty expensive way to make a machine for making ATP, as opposed to doing it on the plasma membrane like bacteria do. But this is how things got started in the last eukaryotic common ancestor. So we're stuck with this legacy forevermore. So mitochondria do produce energy, but on the other hand, they're quite expensive to build themselves. You can look at this in a number of other ways. This basic question about what we get out of mitochondria. I think I probably will just skip over some of this, but again, I guess maybe the main point here is this is a scaling relationship between the number of ATP synthase complexes per cell and the surface area of cells. And it's no different if you were just to extend the data from bacteria up into eukaryotes, you see they fall right on the line where it would be in bacteria. They're not on the plasma membrane, but the number of ATP synthases in cells scales in this universal way that you see with bacteria. You see this universal scaling relationship of ribosomes in cells with cell volume across all these different phylogenetic groups. So these all seem to be sort of universal scaling relationships in the entire tree of life. So unresolved issues on the origin of the mitochondria. I think the data really suggests that rather than launching an energetic revolution, the mitochondrial host cell consortium at best might have been a zero-sum game. And at worst, the eukaryotic host cell was worse off than it was before the colonization of the mitochondria. There's no reason to believe that the primordial mitochondrion wasn't simply an energy parasite that eventually got colonized. In fact, in the phylogenetic tree of bacteria, the mitochondria falls right in the midst of a bunch of intracellular parasitic bacteria, things like the rickettsia and so on. Why do some of these bioenergetic scaling laws transcend the prokaryotic-eukaryotic boundary despite the origin of the mitochondria? Unclear. We can ask, what's the requirement in terms of the origin of the mitochondria? What was the requirement for the evolution of a stable mutualism? That's what the mitochondrial eukaryotic cell host consortium was. It is an obligate mutualism now. But is it true that the partners in this interaction are better off than they were before? So the question always that we ask with respect to mutualism evolution is the mutualistic consortium more than the sum of its parts? And we really don't yet know whether the mitochondria pre or post-dated all the other aspects of the origin of eukaryotic cellular complexity, like internal vesicle transport systems, the Golgi, the ER, and so on. So there's a lot of open questions still with respect to the origin of the mitochondria and what it had to do with eukaryogenesis. But I think one closed question has to do whether it caused a bioenergetics revolution. And to that, I think the answer is a resounding no based on the data.
I have a question.
Yeah.
So the increase in the number of ATP synthase proteins is due to the number of mitochondria also increase, or it's just the surface area of mitochondria increases with the increase in cell size and area of plasma membrane across organisms?
Well, the numbers I gave you are for the total surface area summed over all the mitochondria.
Okay.
I can't tell you off the top of my head how the number of mitochondria would scale. I think probably not in a dramatic way. One thing that might not be known is most mitochondria in many different unicellular lineages don't have that nice, simple bean shape. Many of them are more of like a network and so on. So
Right.
making it even difficult to count the number of mitochondria in some cells.
Right. Okay. 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 Thank
Okay?
you.
Mike,
Yeah.
is there a limit on the size of prokaryotes, and that beyond that size, you have to be a eukaryote to be a single cellular organism?
Well, that's a good question. You know, there's still a lot of organisms that, you know, we're still trying to understand the full tree of life. But as I mentioned, I think at the beginning of last class, that there are these new bugs that have been discovered called Thiomargarita. They're prokaryotes. They get up to be about 2 centimeters long. Now, it's true, we don't see prokaryotes that are a meter long, but you know, there can be bacteria that are much larger than the sort of conventional ones that you think about, like E. coli and so on.
Thank you.
Now, those Thiomargarita are multinucleate. Well, shouldn't say nucleate, but they have multiple genomes scattered throughout the cell. But they're not formally multicellular. Okay, let me.
Sorry, I would also have a question.
Sure.
Is it known whether the potential difference across the mitochondrial membrane is always approximately the same, or if the difference is wildly, could that kind of explain the difference in size of the ATP synthesis? Because if the membrane difference is lower, maybe you need more protons to kind of have the same energy to create one ATP.
Yeah, that's a great question. Unfortunately, I don't have the answer for you, but folks do try to measure this. But of course, those measurements are being made in the laboratory under specific media and so on. So they might not be very representative of, the natural environment in which these complexes evolve. But it's quite remarkable how diverse these C ring sizes are. And I think this is something that bears really looking into, in terms of, it's basically a molecular motor. And if you think of protons as the fuel, there's no question there's differences in fuel efficiency. And then the question is, why is that? Okay. Anything else on this first half? Because now I'll go into starting to think about how we can break down a whole cell's energy budget into its various component parts. So let me go back into share screen. That's not what you want to be looking at. Hang on a second. Let me close my other one and maybe I'll. Okay, here we go. Okay, so we've gained a little insight into how many ATPs it takes to make a whole cell. And now we want to start breaking things down into thinking about how cells apportion their energy budgets into alternative functions. And we'd like to think about how does that fractionation vary among phylogenetic lineages? What kind of traits are easier to evolve in terms of their energetic cost and so on. And this does raise the question, and maybe some of you will have some alternative ideas, is what is the appropriate currency for a bioenergetic cost when you're synthesizing something? And we like to use ATP, count ATP hydrolyses. This is the universal energy currency in cells across the tree of life. All organisms use ATP for most of their bioenergetic transactions. GTP and CTP are occasionally used, but we can make a parallel transformation with those. And then there are other things that get involved, like NADH and FADH. I'm getting into some biochemistry. I don't want to get too deep into the weeds, but there's other things that also can be converted into ATP equivalents because they enter things like the electron transport chain. And then we can go back through that little scheme I showed you about converting protons to ATP and so on. You might argue that maybe we shouldn't be using energy as the sort of baseline measure of the demands on a cell. Maybe we should use something like elemental composition, like carbon. You could certainly make a carbon budget, or maybe some other element. Maybe an organism's limited by iron or something, or you're a diatom and you're limited by silicon. The problem with these elemental approaches is that in some cases, they can be used to estimate the cost of a particular trait, like the shell of a diatom is made of silicon and so on, but they don't easily translate into maintenance or operational costs. So there we really need a measure of energy, and so we use ATP. The next thing that we need to think about is evolutionary biologists, is if we think about these costs of building or modifying traits, how does that relate to fitness, and how can we tie that back into evolutionary theory, which is what we need to have a true, formal theory of evolutionary cell biology. And after going through these basic concepts, then I'll try and give you a series of different applications. The cost of maintaining and operating a gene, how that varies across the tree of life, the cost of membranes. I've already given you a taste of that with respect to mitochondria. We want to think about, we've got a lot of data now on ciliated protozoans, the ciliates, things like paramecium and tetrahymen and stentor, enough so that we can start to think roughly about how the energy budgets are partitioned out. And then I'll show you an interesting universal scaling law in the cost of swimming that seems to apply across the entire tree of life. So the way to start here, the roadmap to sort of way to start thinking about things is that for building any kind of trait inside a cell, there's 4 primary cost components. So, we have to bring in carbon and energy substrates here. So these are our resources that we're bringing in. So there's going to be a cost of acquisition, whether that's transporters in the cell membrane, whether you're a predator swimming around, hunting for food or so on. There's some baseline cost of just getting nutrients. From those nutrients that we get, say glucose, those are broken down into what we call precursor molecules. I'll give you examples of those. And then those precursor molecules are modified subsequently to give the various monomeric building blocks inside cells, and that consumes ATP. So by monomeric building blocks, I'm going to be referring to things like the various amino acids, the 4 different nucleotides or ribonucleotides, and various lipids. So those are sort of the bricks, the basic building bricks upon which cells are built. But then those monomeric building blocks have to be pulled together. You have to glue your amino acids together to make proteins and so on. So there's also assembly cost. And then finally, once you've got your trait or your protein or whatever it is, there's going to be maintenance cost throughout. So when we think about a trait, we would like to sum up all these different sources of demand. But there's one additional thing that we need to think about, and this is what we call the opportunity cost. So imagine you have a glucose molecule, and one thing you could do is just squeeze all the ATP out of it you could by bringing it all the way down through glycolysis, the Krebs cycle, and so on, and you'd get about 32 ATPs. But if on the way down you take a precursor molecule and you no longer use it for energy extraction, but use it to build an amino acid or a ribonucleotide, then you lost that opportunity to gain energy. So there is energy in the original substrate that you're using, but now it remains tied up into the precursor molecule and the monomeric building block you're going to make. So I'm going to go through some probably fairly quickly, just to give you a rough idea of how we estimate the cost of making building blocks in a second. But the idea, again, is fairly simple here. We're going to start with a carbon energy substrate. We're going to ask how much does it cost to make an amino acid or a ribonucleotide or a lipid molecule. And as again, you go through a precursor molecule, which I'll show you in a second, and then you modify that by using ATP equivalents. And again, there's this opportunity loss. And we sum those together to get the cost of a total building block. And I'll go through a couple of examples. I don't want to freeze your brain with a lot of biochemistry, but I want you to get an idea where some of these things are coming from. But just as sort of the big picture here, there's 20 flavors of amino acids, and on average, the cost of synthesizing an amino acid is equivalent to about 30 ATPs. I'm just trying to give you the broad picture here. Nucleotides are a bit more expensive than amino acids. The biosynthetic cost of a nucleotide, on average, there's 4 flavors, of course, and we'll see they're quite similar to each other. It's about 50 ATPs. Lipid molecules, which we make our membranes out of, those are quite expensive. So you want to be careful about not over-investing in lipids if you don't have to. And then just as one more example, peptidoglycan. This is a complex of molecules that bacteria make their cell walls out of. And the subunits of peptidoglycan are also quite expensive. So the important point is you can make this big list of how much it costs to make all the basic building blocks in a cell. And then if you know what your trait is made out of, you know, how many amino acids, for example, you can calculate the cost of the entire trait. So the way we do this is some of you have most of you have probably been terrified at one point or another taking a biochemistry course and having to be confronted with the glycolytic pathway and the TCA cycle, also known as the Krebs cycle, also known as the citric acid cycle. But these are basic textbook pathways that you can find in all biochemistry books. That's the main point. Biochemists have studied all these intervening steps in excruciating detail, and they know all the biochemical reactions step by step. Each step is just usually some single modification. So here we start with glucose, and then we actually add a phosphorus to it. So this is actually more energetically valuable than glucose is. But then the way down, now we break the 6-carbon sugar into 2 3-carbon products, and you go down this pathway and so on. And going down this pathway, we're squeezing out some ATP or NADH and so on. And then we get down, passing acetyl-CoA, pyruvate down into the citric acid cycle. And there's quite a few components of this cycle that I'm not showing you here. But the main point here is that we really understand what's going on in making all these precursors as we go down this table. And this will become important because these end up being the precursors used to make different amino acids and so on. These guys over here on the pentose phosphate shunt, these precursors are used in the production of nucleotides. You can see there's NADHs and sometimes FADHs are used, but we can convert those to ATP equivalents with these sort of benchmark estimates here coming from basic biochemistry. And the book chapter that I pointed out yesterday sort of gives a series of recipes for getting everything you need here. We put it's not fun to do this kind of thing unless maybe you're a biochemist and you really enjoy it. But the main point is you can squeeze this information out of our basic understanding of biochemical pathways, and they're mostly quite conserved across all of life. So you can see these abbreviations here. They match up to these precursors over here. And then we can figure out how much ATP is used in getting there, NADH and FADH. And by using these conversion factors, now we can get a measure of the value of these different precursors in terms of their ATP equivalents. So if you totally bypass that, that would be your opportunity loss if you weren't squeezing any more energy out of it. So I have these tables for the value of the precursors. And just to give you one example, let's think about how much it costs to make the 4 nucleotides. You start with, say, glucose. You make G6P. This PRPP in the pentose phosphate shunt is worth 30 ATPs. So right there's a cost because you're pulling this off to make your nucleotides. If you're making a T or a C, you have to pull in an aspartate. If you're making a G or an A, you have to pull in glycine. This is the biosynthetic cost of glycine, biosynthetic cost of aspartate. And there's various other steps. We go down these pathways. These are things you can always look up. You don't need to memorize this, but I'm just trying to give you a flavor of how you get the final biosynthetic cost of the four different deoxyribonucleotides. They're down here. And I've given them up here again, up to this point here. And the main point I want to make is most of the cost of a ribonucleotide is opportunity loss cost. And then there's the direct, and the sum of these is the total cost. And just to give you an idea what this can start to tell you, if we sum up an A and a T, we get 100.5. And if we sum up a G and a C, we get a 98.5. So if all that mattered to life was trying to minimize the cost of DNA, there'd be a premium on using GCs in your codons as opposed to AGTs. It's not a great difference, but it's nonetheless a difference. Yet most organisms seem to have a mutation bias, it turns out, towards A's and T's, the most expensive nucleotide pair.
Mike,
And then we can similarly think about what about amino acids?
Mike,
Yeah.
I have a question. Yeah.
Sure.
So is there anything known about the total number of ATP molecules in a cell at a steady state? As in the cell may be, making up and breaking down, and constantly ATP is being made and utilized. But at a given steady state, is there anything known about the numbers of a ATP pool, ATP reservoir, or something like that?
Great question. It's difficult to measure ATP directly. Is there anyone in the room who has an answer to that one?
I think the concentration is very, very large. It's much more than you need.
Much more ATP than you need. It's a large number.
Millimolar, you said?
Pardon
1
me?
to 10 millimolars, they suggest me.
Okay.
So it's very large. That,
Right. Okay.
But of course, it's turning over, and that's what we're sort of after here, right?
Okay. Thank you.
Okay, now to move on to amino acids. Remember, I just showed you the glycolytic pathway. Hopefully you can see my pointer. Yeah.
I'd just like you to clarify what exactly is opportunity cost. I didn't quite get it.
Maybe I can do it with this diagram here. So we start with glucose. That's worth, if you were to do nothing with glucose, but squeeze every last drop out of it just to make ATP, to extract energy, you would get about 32 ATPs in most things, I think. But suppose you're starting with glucose and you want to make serine. You have to take this intermediate 3-phosphoglycerate here, and that's a substrate for making serine. So that's no longer available to extract energy. So this is sort of an economics term that's frequently used as opportunity cost. So you lost the opportunity to get more energy in making this particular molecule for the cell. Does that make sense?
Yeah. Thank you.
So we're trying to account for this opportunity cost because it is energy contained in the original glucose molecule. So while we're at it, let's think about the biosynthetic cost of making amino acids. Again, anyone who's got a biochemistry textbook is probably and has looked at it has probably seen these kind of pathways. Here's the glycolytic pathway again. I just showed you that. Going out in the TCA cycle in more detail here. And these are the various diversions off these different precursors to make all the amino acids that we require in the cell. So all of us, eukaryotes and prokaryotes, require 20 amino acids. So these are the biosynthetic pathways for the various amino acids. So we know the cost of diversion of a precursor, And then we know what all these pathways are. There's quite a lot of details not shown here, but these are in all the textbooks. And you can then figure out the cost of making various amino acids. And that's what I've summarized over here in this table. On average, the opportunity cost for amino acids, 24 ATPs. So you're sort of leaving ATP equivalents behind in the substrate that you're using. And then there's these direct costs of on average 6 ATPs that are used in actually converting to the next step and the next step and the next step. And then the sum, then, the average amino acid, as I pointed out before, has a total cost of about 30. But unlike nucleotides, there's quite a bit of variation among the cost of different amino acids. So, for example, I think glycine is at the bottom of the heap here, 12 ATPs, but then you get into tryptophan, 71. So there's a real difference in the cost of biosynthesis of different amino acids, which will raise the question, and we'll try to answer that. Is there a priority in different organisms of utilizing different amino acids in the protein based on their energetic cost? So all this is, excruciating amount of work. They have to, to squeeze all this information out of these elaborate biochemical pathways. The beauty is that biochemists have been working on these kinds of things for years, and we just really know the details. So this can be done for common molecules like amino acids and nucleotides, but there's lots of other organic molecules inside cells that we don't necessarily know the pathways, the biosynthetic pathways, and the pathways might even differ among different organisms. That's actually true for the pathways that I've been talking about up to now. There are variants in different lineages. There can be non-orthologous gene replacement in the genes involved in these different pathways, but that will end up giving us about the same results. But we recently discovered there's a remarkably simple way of calculating the cost of a biomolecule, one of these building blocks that I just mentioned, without any knowledge of the biochemical pathway. And what I've illustrated here is results for amino acids, various precursors, nucleotides, and wall and membrane components, which I'll talk about in a minute, lipids and peptidoglycans. We know what these costs are based on biochemistry. That's the values on the y-axis here. There's this old work in physical organic chemistry. There's a whole book written by Karash and Scher almost 100 years ago. And this book measures what's called the heat of combustion of every organic substance you can imagine. So this is what chemists like this do. They just burn a substance and figure out how much heat comes off. That's the heat of combustion. And these guys pointed out that the heat of combustion of an organic substance is very closely related to the first three terms here. You just take the biochemical expression for your substance, you count the number of carbon atoms, multiply by 4, count the number of hydrogen atoms, and subtract from that the number of oxygen atoms multiplied by 2. And this is what they call the degree of reduction. So more hydrogen, you're a more reduced compound, and more oxygen, you're more oxidized, and you can squeeze less energy out of it. But they pointed out that this simple counting mechanism is a great, very accurate measure of the heat of combustion in a substance. And then we found that if you add in 10 weight each phosphorus in your substance, if it happens to have phosphorus, this wouldn't be the case for amino acids, but that gives you these values here on the x-axis. And the point here is that these are very, very highly correlated. This is a log-log plot, so this works really well over orders of magnitude different range. It's a very simple way to predict the cost of making various building blocks inside cells, and you can you can extend this to totally different kinds of organic molecules, things like osmolytes, all kinds of things. So I think this opens up a real opportunity to, no pun intended, a real opportunity to calculate the cost of building various things inside cells just based on their organic composition, their elemental composition. Okay?
I have a quick question.
Sure.
I'm not sure I understood. Is this an empirical law or is there a way to justify it more or less formally?
Well, it was originally an empirical law, but I guess the way you can think about it, this sort of makes sense, right? So each carbon has 4 bonds it can make, I guess. And so you can think of this in that, excuse me. But it's fair to say it started out with these guys as an empirical relationship. But I think I'm not a good enough chemist to actually sit here and derive this, but hopefully the basic concept makes sense. So if you had CO2, that's 1 C, that's a 4, and 2 O2, so that's a -4, and the heat of combustion would be 0 for CO2.
Okay, thanks.
But you might want to do some additional reading on this, but I mean, it is based on in general organic chemistry principles.
A question.
Yeah,
So this is cost in terms of energy.
yeah.
Should one also be associating other costs for every molecule, like cost in terms of matter, how much carbon it has? Or is this all in terms, I mean, does it take care of everything? Or are there other costs that one should worry about?
Yeah, no, that's a good point. That's what the point I was trying to make earlier. Because we're interested in sort of universal scaling relationships across the whole tree of life. We need a universal measure of currency. So that's one reason for using ATP. You could use carbon. Everybody needs carbon. And then if you go, so there's no reason you couldn't do everything that I'm talking about today using some kind of carbon budget, I guess. But even there, it becomes difficult to, that works when you're doing the building cost, the cost of building things, but the cost of maintenance becomes much more difficult. I'm not clear how you would use carbon equivalents for things related to respiration or just basic repair of enzymes and so on. So that's the reason we've stuck with ATP, but in principle, all these things could be extended to different elemental compositions if you like. One amazing feature of biology is, there's what, 100, and I don't know what we're up to now, like 114 known elements. Biology only uses roughly 20 elements out of the whole elemental table. Okay, so that's how we get these basic costs. And we want to relate this now to evolutionary theory. So we want to have an evolutionary cell biology. We need some basic fundamental theory for converting these kinds of observations into evolutionary currency. And so the way we can think about this is think about the cost of a gene, and I'll go through a specific example here. It's going to have a cost at the DNA level. It lives on chromosomes. At the RNA level, the gene is transcribed into RNA and then eventually into the cost of proteins that you want to make. And so you can think about that as the total cost of a gene without respect to whether the gene is doing anything for you whatsoever. Now, that gene might be doing something to you. And if you're an ecologist, you might measure the direct selective advantage of having that gene. Maybe it helps you survive or something like that. But the net selective advantage of the trait, we really should be subtracting the cost of building the trait from the advantage that you get out of it. And if these are the same, it's basically a wash. It doesn't matter whether you have it or not. So the way to think about it is we start with this is just a simple scalar here. So we start with a situation where the gene is absent, and then we add the gene to our bug, but it's not doing anything for the bug. It's costing the bug, though, because of all these biosynthetic costs at the DNA, RNA, and protein level. So there's a baseline cost of harboring any kind of trait in any organism. And then you can imagine, well, let's suppose there was no baseline cost, and ecologically, this is the benefit. This is what you would get out of having the trait. And the net advantage is the difference between these. This is what we'd like to know ultimately. Today, we'll be focusing just on the baseline cost. Okay, so this would be the net advantage of a trait or a protein or whatever after subtracting out the cost of building and maintaining it. So that's how I think we can start connecting with evolutionary theory. And always we have to scale these costs of any trait, any protein, whatever it is, to the total cell energy budget. Because if the cost is X and the total cell energy budget is 10 to the 9th times X, that's a small cost compared to the total energy budget was 10 to the 4th times X. So recall yesterday I showed you various ways we can estimate that we now have some pretty good estimates of the total ATP demands of a cell. So that's the denominator I just made. We know the total cost of a cell, and then we want to convert this into a selective advantage or disadvantage of adding a new trait to a cell. So the way to think about this is we're thinking about this without yet accounting for, we just want to know the baseline costs. We're not yet accounting for any advantages that might be accrued. And so there's going to be some baseline costs. This will be a reduction in the growth rate of individuals harboring that modification. The fitness before trait modification, we just use a benchmark of 1. This is how things work in evolution. Everything's relative to each other. So you always benchmark the time 0 situation is 1. And then the fitness after the investment in the trait is 1 minus the selective cost. The cell has a total energy budget of big C, and little c is the added energy cost of adding this trait into the cell. So the way this works is the ancestral cell division time will be proportional to the total cell's energy budget. If your energy budget is doubled, then it's going to take you twice as long to accumulate the energy for the cell to divide. And then if we add this modification to the cell, the division time after the trait is big C plus little c, the added energy cost, total energy cost of the cell. This is important because this is the currency upon which natural selection operates. Longer division times means you enter your genes into the gene pool at a slower rate, and you'll fall behind. I won't go through the math here, but what things work out to be is that the selective cost of a trait turns out to be simply the ratio of this added cost relative to the total cell energy budget. So that should be almost intuitive, but there's a fair amount of math that goes beyond this. That's the answer. If your organisms were reproducing in discrete generation, so a generation followed by everybody reproduces the next generation and so on. If you have overlapping generations, this ln of 2 comes in. So that's not a slight difference, but that's where this comes in. That's what about 0.69 or so. So you get, it's probably simplest to just think about this ratio, the cost of your trait relative to the total cell's energy budget before you have the trait. So that's something we can measure. So let's think about this with a particular application now. There have been many arguments made over time, often by people sort of in the field of ecology or other related fields, arguing that selection can promote amino acid compositions of proteins based on the differential cost of those amino acids. So we want to now think about whether this is really possible in evolution, given the theory that I just mentioned. So the way to think about it, the simplest way to go at this is, let's think about the maximum cost differential between two amino acids. Glycine's the cheapest, tryptophan is the most expensive. The difference in biosynthetic cost is 59 ATPs. And we want to ask, would a cell be able to even discriminate these two amino acids from each other? Because if it can't, then it certainly can't discriminate two others that have a smaller differential. So based on that plot I just showed you, the cost of building a whole cell is about 3 times 10 to the 10th ATPs times the volume of the cell in cubic microns. So that comes from that regression that I showed you a few minutes ago, that I also showed you in yesterday's talk. So that's the cost of building a cell. And now let's do the math for an E. coli cell. So E. coli conveniently has a cell volume of 1, so we have a 1 over here. And now let's think of a highly expressed gene, because this is where the advantage of using glycine over tryptophan might be greatest, because there's a lot of the proteins in the cell. This is a change at one particular amino acid in the protein. But for an E. coli, a highly expressed gene would have roughly 10 to the 4th proteins in the cell. And what we want to do is compare this particular protein at one amino acid site where there's a glycine versus a tryptophan. So the differential will be 59 times 10 to the 4th. Right? We have 10 to the 4th proteins, and one of them has glycine and the other has a tryptophan in a different mutant. And that's the total cell energy budget, 3 times 10 to the 10th. The relative cost then is 2 times 10 to the minus 5. So that would be the advantage of not using glycine over tryptophan, or the selected disadvantage of a tryptophan got substituted for a glycine. So this gets us back to our evolutionary theory. For selection to be effective, the selection coefficient has to be greater than the reciprocal of the effective population size. In other words, this quantity here, 2 NES, has to exceed 1. So for selection to be able to discriminate something like glycine from tryptophan, NE and E. coli would only have to be, the solution here is plugging this S into here and rearranging to get NE. NE would only have to be 4 times 10 to the 4th. That's a pretty small effective population size for bacteria. E. coli has an effective size around 10 to the 9th. So E. coli, really, natural selection operating in a bacterial population like E. coli really would be able to discriminate these two amino acids. There could be selection just based on the bioenergetic investment in 2 different amino acids like this. As I said, this is as good as it gets, because these are as far apart on the spectrum as you can be. But for a highly expressed gene, E. coli could make that discrimination. Now, I'm not saying anything about the functional aspects of glycine or tryptophan. They might also influence how the gene operates, but this is just a bioenergetic question. Now let's switch to a lowly expressed gene with only 10 proteins, and we have to divide everything by 1,000, and then the critical NE is about what is that, 400 million. And that's still within the realm of possibility for bacteria. So this is telling us that in bacteria, there really can be selection for amino acid composition in proteins just based on the differential biosynthetic cost of the different amino acids. Now let's think about a couple of other eukaryotic cells. For yeast, a yeast cell has a volume about 100 cubic microns. So that means the lifetime cost of a cell is about 100 times more than it is for E. coli. And without going through all these numbers, the critical effective population size for low and highly expressed yeast genes would be about 4 times 10 to the 8th or 4 times 10 to the 4th. So this would work for highly expressed genes. This is where you'd have to be for lowly expressed genes. And yeast have an effective size somewhere around 10 to the 7th or so. So this is telling us that For highly expressed genes, there could be selection for differential amino acid utilization in yeast, but not for lowly expressed genes. The penalty you would be feeling would just not be as great because you're just not harboring as many proteins with those alternative amino acids. Then we jump into an animal cell. This is sort of a moderate sized animal cell, 1,000 cubic microns. And then these numbers jump up to the ones down here. And now we're getting to the point where in a mammal or a vertebrate, probably a land plant in general, effective population sizes just are never large enough to discriminate amino acid utilization and proteins based on their bioenergetic capacity, unless the gene is really enormously expressed in the cell. So the point here is we can use these kinds of bioenergetic arguments to start thinking about what natural selection can and can't do in different phylogenetic lineages. So now let's think about the cost of a whole protein coding gene. So the example I just gave was just for one amino acid substitution, but now we want to think about the cost of a gene. Bacteria typically have 4,000 protein coding genes or so. Us vertebrates have something more around 20,000 or so. And most organisms should be frequently in a use it or lose it situation. We want to think about when you're in a situation where the burden of a gene that's not paying itself back in terms of fitness is so great that selection will promote a deletion of the gene. So the cost of a protein coding gene, there's 3 levels of cost. There's a cost at the chromosome level. We have to synthesize the nucleotides. They have to be glued together in chains, double-stranded DNA. And then there's a few downstream transactions that are involved in modifying nucleotides and so on. There's a cost at the transcription level. We have to synthesize mRNAs from ribonucleotides. And for transcription, we have an additional problem that most transcripts only last a few minutes inside cells. So we have to be continually building more transcripts inside the cells. We have to account for turnover of transcripts required to maintain a steady state. And at the protein level, we have the same thing. We have turnover of proteins, although this is much more slow than it is for mRNAs. But again, we have to now account for the synthesis of all the amino acids that are strung together in the proteins. There can be downstream modifications of proteins and so on. And we want to sum all these 3 levels of cost together in terms of ATP hydrolyses equivalents. And so the total cost of a gene is the sum of these 3 costs. And the gene might be doing something for us. We're not going to talk about that today. We just want to think about the baseline cost of just maintaining and operating a gene. And this becomes important because if that cost is less than the power of random genetic drift, then selection would be unable to eradicate the gene, even if it wasn't paying for itself.
Mike?
Yeah.
Does it seem that in bacteria there's more or less glycine than tryptophan? Or is that, has evolution chosen for glycine or tryptophan across bacteria?
Yeah, there is some evidence of that in bacteria. A number of years ago, Hiroshi Akashi published a paper, I think it was in PNAS, maybe like 20 years ago, and they were looking at this sort of thing in bacteria. And so as I recall, in bacteria, there is this, I mean, you can't give up tryptophan entirely, of course, because it sometimes has functional significance. But there is differential utilization of the cheaper amino acids, especially in highly expressed genes in bacteria. So that makes it consistent with the arguments that I was just making.
Thank you.
Okay, so let's now break things down in the cost of a gene. I just want to give you an idea how we go about getting these numbers. So at the chromosomal level, the primary cost is going to be in nucleotide synthesis. Remember, I told you that it costs about 50 ATPs for every nucleotide. DNA comes double-stranded, so I have to multiply by 2. So there's 100 ATP equivalent costs per site, and then we multiply by the length of the gene and the number of base pairs. Now, in eukaryotes, there's an additional big cost at the chromosome level, and that's because we wind our DNA around these protein complexes made of 8 proteins. These are called nucleosomes. They're wrapped around a complex of histone proteins. There's also a linker protein. That brings in an additional cost equivalent to 160 ATPs per base pair across chromosomes, okay? Regardless of whether there's a gene there or not, that is the cost of histones. So that's comparable, if not more, than the cost at the DNA level. So that's a considerable added expense that we have at the chromosome level in us eukaryotes. There's a lot of other costs that require energy at the DNA level. You want to replicate your DNA, it takes energy to open the origins of replication, it takes energy to unwind the double helix. There's these little RNA primers that get things off the ground in replication. You have to ligate the so-called Okazaki fragments in replication and so on. These are all not zero cost, but they're out in the second decimal place compared to these other 2 costs that I've promoted here. So they can be more or less ignored. And when you put everything together, you have these simple expressions for the cost of a gene in bacteria at the DNA level. It's 101. There's 100 up here, and all this other stuff adds it to about 1 times the length of a gene. In haploid eukaryotes, this is quite a bit higher, and that's because we're adding in all this extra cost due to nucleosome. And then in a diploid, this basically doubles up with respect to haploids. So the cost of a gene at the DNA, at the chromosome level, is considerably higher than it is for bacteria because of our use of nucleosomes. There's also big differences. I just multiplied here by the length of a gene, but the length of genes in eukaryotes is also quite a bit longer. Now, what about at the transcriptional level? Ribonucleotides are a little bit cheaper. Instead of 50, we use a 46. We need to know the steady state number of mRNAs per gene in the cell. This is just a baseline synthesis for the mRNAs that are sitting in the cell at any one point in time. And then we multiply by the length of the transcript in bases. But we have to worry about one additional problem, and that is messenger RNA turnover inside the cell. So the way you can think about this is cells have sort of a steady state number of mRNAs in them, but they have a decay rate, something like a half rate of typically just a few minutes. And so the product of these, to keep steady state, the product of these must be the rate of production of new, this is the death rate of transcripts. So this must be the birth rate of new transcripts to keep things at steady state. So we have to bring these into the final equation. We assume that ribonucleotides are recycled, but every time you make a new mRNA, you still have to invest 2 ATPs to string things together and new transcripts. In eukaryotes, there's an additional cost. We put poly-A tails of about 100 base pairs in length. So that's an additional cost that bacteria don't have to pay. And there's a bunch of other costs that can come in. But again, these tend out to be secondary or even tertiary. There's cost of splicing in eukaryotes. We have introns, costs energy to splice those out. There's cost of histone remodeling and so on. Bottom line, when you put all these together, you again get these fairly simple expressions for the cost of a gene at the RNA level in bacteria and eukaryotes. It's a function of the number of transcripts in the cell at steady state, the length of those transcripts. This is the decay rate. And this is the cell division time. So in effect, that product there is the number of replacements that you have to make in the lifetime of a cell. So if you know the decay rate, the length of the gene, cell division rate, and you know something about the number of transcripts in the cell, we can calculate the cost at the transcript level. And then finally, there's the cost at the protein level. The idea is the same again. There's going to be a cost of each amino acid costs a certain amount. I showed you that table. There's a step, and the cost here is going to depend on the amino acid composition of your protein. If you know the number of proteins per gene and the length of the protein, now you know the cost of amino acid synthesis for the protein. The ribosome has to glue these proteins together, and each additional amino acid that's glued to the preceding chain, that costs 4 ATPs per residue, so we have to take that into account. We also have to account, again, for the turnover due to protein decay in cells. And there's a small number of other things that are involved. Again, they're out in the second, third decimal place. And you end up with this simple algebraic expression for the cost of the protein level. And again, you might not follow exactly the math here, but it should be intuitive. The cost of the protein level is proportional to the number of protein molecules in the cell, the length of the protein, the number of amino acids in the protein, and it's a function of the decay rate and of the cell division time. So if their proteins didn't decay in cells, this whole second term would disappear. Usefully, over the past few years, people...
Mike, I have a question.
Yeah, yeah.
In the cost of protein, shouldn't we account as well the cost of the ribosomes that produce that protein? So if you want to produce more protein, you should produce more ribosomes, right?
Right. Yeah, in principle, that could be brought in. Not sure how you would do that. You would have to average it over all the proteins in a cell. I mean, those expressions that I just showed you could be used to calculate the cost of making a ribosome, for example, itself, right? A ribosome has maybe 100 proteins in it, and you can apply those results to each one of those proteins, sum them up, and that's the cost of making a ribosome.
I guess the way I'm thinking about it is that you probably want to have a certain ribosome to other protein ratio. And so if you want to do more protein, you need more ribosomes, otherwise you can't grow at the same rate. So I don't know if that's in the same order or, yeah.
Yeah. Well, I'm really not sure if it's appropriate to add those costs to the cost of each individual gene. I'd have to think about that a little more.
Okay.
I think I understand your point.
Thanks.
Okay, so I just gave you these expressions, but you know, we need some information. We need to know how many proteins are in a typical cell. Getting the length of a protein or an mRNA is usually not that difficult in the age of genomics, because you know we have whole genome sequence and we know what that is. Getting decay rates is a little harder. The way that's usually done for decay rates, say in mRNA, is you poison the cell so it can't transcribe anymore, and you just follow the cells, and you see a dissipation of the number of mRNAs in the cell over time, and that is thought to give you a measure of the decay rate. Similarly for proteins, you poison the ribosomes. That's one way that people try to get at those. But what I'm showing you here is some more of these interesting universal scaling relationships. So the top plot here, we're looking at each one of these sets of points is for a different species in which measurements have made of the proteome, the number of proteins for each number of protein molecules for each protein coding gene inside the cell. And the upper line is just the total number of proteins summed over the whole proteome. And these are the mean number of protein molecules per gene. And I think this lower one, I think, is the median, as I recall. But you'll notice there's this nice scaling relationship with cell volume. And these are really different organisms ranging from eukaryotic cells down to prokaryotic cells. So again, this suggests at least a crude way, if you know the cell volume of your organism, you could use these regressions to estimate the mean number and the total number of proteins per cell. And similarly, unfortunately, we have less data for messenger RNAs, but you can again see these general scaling relationships. So the point here is if you know the cell volume, you can make a pretty good estimated guess of the average number of proteins produced per gene. What I've done over here is I said, let's follow different points here and take the mean. I've done this for different organisms here. Mycoplasma is a very small bacterium. E. coli, you're all familiar with. This is budding yeast, Saccharomyces, and this is for a mammalian cell. And what these are just showing you is the probability distribution for the number of proteins over the whole proteome. So, smaller cells, the mean is lower, and the variance is actually somewhat reduced, and so on for proteins. The point to make here is that, these are hugging a value of 1. So some proteins are only transiently inside a cell, whereas others, in a mycoplasma, the average is only about 10 proteins per protein coding gene. So there's a lot of stochasticity going on inside cells. This is for mRNAs, and this gray area is where you're below 1. So many genes inside even small eukaryote, moderately sized eukaryote cells will transiently go through a period in which there's no mRNA in the cell. And then you have to wait for the gene to fire again. Proteins have a longer half-life, and so they're more guarded against being lost transiently in the cells. The main point is we have some information now on the number of proteins of every gene inside a cell in certain species. And that's going to enable us now to think about, for some model species, the cost of the whole set of genes in the species. So not surprisingly, one of the bugs that we have all the data needed to make these calculations is E. coli. What I've done in this plot, these are histograms, really. These are the frequency distributions of the cost of the some 4,000 genes in E. coli at the replication level, that's the chromosome level. So at the DNA level, average gene is costing about 10 to the, this is log scale, about 10 to the 5th ATPs per cell cycle. The orange here are the costs at the transcription level, the yellow at the translation level, and the blue is just the sum of all. It's the histogram for the total cost of a gene. So the total cost of a gene is something like, I don't know, about 10 to the 5.5 power or something in E. coli. So those are the absolute costs. Now remember, what's important here is we have to take the absolute cost of a gene and divide it by the total energy budget of the cell, the total cost of a cell. And that's what's done up here. We're just dividing all these numbers down here by 3 times 10 to the 10th, which is the total energy budget of an E. coli cell. And so these are the fractional costs of the cell's energy budget. And then finally, there's these dash bars over here. This is what I talked about yesterday. That's the drift barrier. So this is 10 to the minus 9 here. E. coli has an effective population size of about 10 to the 9th here. So anything that resides to the left of here, natural selection, if you were a gene living over here, not doing anything for the cell, natural selection would not be able to promote a deletion of the gene. If you reside to the right of this, selection can see everything effectively in E. coli. And you can see essentially all E. coli genes. These are probably just some misannotated genes in the genome. But in E. coli, almost everybody lies far, far to the right of the drift barrier, orders of magnitude. So effectively, from a bioenergetics perspective, selection could see anything in E. coli. Remarkably, an insertion of only a couple of base pairs in E. coli has one base pair, remember, is 100 ATPs. And that can almost be felt by natural selection. So I think these kind of results really help explain why bacterial genomes are so streamlined. Bacterial genomes that consist of about 95% of coding DNA, very little intergenic DNA, very little, if any, transposable elements, and so on. Any kind of insertion like that that's not paying for itself in terms of fitness is easily discriminated by natural selection in a mutant containing that variant. So now let's move up the ladder, so to speak, two other model systems, yeast here, and then famous plant, Arabidopsis thaliana. There's three things that happen in these plots. So yeast is a famous unicellular eukaryote model system land plant. And what happens is that these plots here move to the right, scale changes in one of these cases. So the cost of the gene is increasing for a variety of reasons. Histones that bacteria don't have to pay for. Some genes, especially in land plants and vertebrates, contain lots of introns. That's extra DNA you're paying for, and so on. So the absolute values of these move to the right. On the other hand, the cell size is much larger. So the denominator is becoming larger. The relative costs are becoming smaller. Relative costs of the gene are smaller because the total energy budget is larger. And then the drift barrier is moving to the right because these organisms have smaller effective population sizes than do bacteria. And you see what gradually happens is that most of these distributions are falling to the left of the drift barrier, a good proportion of them. And that means that if a gene stops paying for itself in terms of fitness, selection is not going to be able to eradicate it. It's not going to be a promoted deletion. It might drift out of the population, but it's not going to be promoted by selection just because natural selection, the eyes of natural selection are effectively glazed over. So these absolute costs, as I just said, are 10 to 100, absolute cost of a gene is 10 to 100 times that in a bacterial gene, but the relative costs are smaller because the cell volume is larger. And as an example, in Arabidopsis thaliana, there's an entire mitochondrial genome sitting on one of the chromosomes. Somehow it got embedded in one of the chromosomes. It's not doing anything there at all. It's about 30 kilobases in length. That's a lot to be paying for DNA, but relative to the total cell's energy budget, it can't be felt. So the main point here is we can use all these approaches to start asking, evolutionary genetic questions. Why is there genomic streamlining in some species, not in others, and so on. So I just repeating myself, the energetic cost of just a few nucleotides is sufficient to be perceived by natural selection in bacteria, but even large insertions are effectively neutral in eukaryotes. Generally speaking, the costs at the RNA level exceed those at the DNA level, not always, but often so. RNA is single-stranded, DNA is double-stranded, so that's an additional penalty of DNA. But highly expressed genes, you're paying more at the DNA level. But most of the cost of a gene in most organisms at the protein level because of the large number of amino acids and large number of protein molecules in a cell. And what all this is telling us is that some people in the past have argued that increased cell size imposes a burden on the colonization of excess DNA or excess genes. But the argument here is that it actually makes it easier for the genome to be colonized by excess DNA because the fractional contribution is diminished. All right, moving on to lipid molecules in membranes. I'm not going to go through the details, but I've already pointed out that lipid molecules are much more expensive than amino acids or proteins. So these are just some censuses. There's some species where you have a complete survey of all the different flavors of lipid molecules in the membranes throughout the cell. There are many, many different kinds of lipid molecules. They have different head groups, different tail lengths, different degrees of saturation of the carbon atoms and the tail links and so on, but you can use some of these recipes that we put together to calculate the total cost of a lipid molecule. And they're on the order of about 320 up to almost 400 ATPs per molecule. So much higher than nucleotides. Remember, a nucleotide costs us 50 ATPs. And yet, even though different species have different types of lipids they're using, they're all in sort of the same ballpark. And even within eukaryotes, the cost of an average lipid in a plasma membrane is not that much different than the cost of an average lipid in the mitochondria. And they're slightly different molecules, but the overall cost is about the same. And then we can start to think about the cost of membranes inside cells. And with a certain amount of information, this is fairly easy to do. We have the cost of a membrane is equal to the total number of lipid molecules in the membrane times the average cost per molecule, which I just gave you. To get the total number of lipid molecules, we have to multiply the membrane surface area by 2, because lipid membranes are bilayers. There's 2 juxtaposed layers with tail-to-tails with their hydrophobic tails paired up against each other. So we multiply by 2. We know how much area an individual lipid molecule takes up. We know that the so-called headspace per lipid molecule. So we divide the area by the space each lipid molecule takes up. That gives us the total number of lipid molecules. We multiply the cost per molecule. We get this kind of expression here. And then we can start to apply this to real data. Over the past few years, people have been starting to do sort of the measures of the internal anatomy of cells. This is an example of some reconstructed internal cellular anatomy of the tiny green alga called Ostriococcus. So there's the nucleus. It's a flat sort of cell. I guess you would call this oblate spheroid, or you get the point. It's a spheroid of some sort. There's a single chloroplast surrounded by a membrane there. There's the mitochondria in here. I think there's 1 or 2 mitochondria. And there's the ER and the Golgi and so on. And the point is, if you have these stacked photomicrographs, you can calculate the total amount of lipid area associated with each one of these organelles. And then you can start to figure out how much of the cell's energy budget is allocated to these different organelles in terms of the lipid membranes involved. And there's only a few organisms for which we can do this. And I've tried to give you a rough idea of how energy budgets portion up. So this would be if we look at bacteria, we're just talking about the plasma membrane. There's none of these organelles here. And this is telling us that the fraction of the total cell's energy budget going into lipid membranes, it's on the order of 20 to 30 or so % of the cell's energy budget, something I pointed out yesterday, the so-called Realtor's law. And then when you get into eukaryotes, we can start to break things down. So here's Ostriococcus. Most of the lipids are in the cell, the plasma membrane, about 3% in the mitochondria, about 15% in the nuclear envelope, and so on. And in Ostriococcus, about 60% of the cell's energy budget is associated with lipid membranes, about half of which or so is associated with the internal membranes. So we pay a penalty in terms of internal membranes and the expense of all the lipid molecules that are used. So again, about 20 to 60% of the total energy budgets of cells is associated with membranes. The numbers are much higher in eukaryotes. And they become higher and higher in cells that are larger and larger. So we can, let me remind myself of where I'm heading. Yeah, I just want to give a couple final examples here. So I've showed you this plot before. We can start to estimate I want to give you just some idea how much mitochondria cost again. And sounds like I already did this, losing track of where I was. So here again is a scaling relationship between the total membrane area of mitochondria in all kinds of eukaryotes of different flavors, unicellular, heterotrophs, mammalian cells, phototrophs, and so on. We have this nice scaling relationship. We can also do the same thing with the nuclear envelope. That scales with the square root of cell volume. But again, there's these scaling relationships, so we can start to write down general rules for how investment in different cellular features changes with the evolution of a change in cell size. When you put all this together for mitochondrial membranes, again, about 5% independent of cell size, about 5% of the cell's energy budget goes into just maintaining mitochondrial membranes, just the lipids themselves. It's not including the proteins that happen to be in the membranes. And then this is just sort of there's a lot of information here, and it's not important that you go through it, but I'm trying to point out that for some organisms like ciliated protozoans, like this paramecium, would include tetrahymen and other model species, we're now able to break down the whole energy budget into different components. And just as an example, these are the fractions of the cell's energy budget for cells of different cell volumes. So these are the smallest known ciliates, and these are the largest here. And then I've worked them out specifically for tetrahymen and paramecium. And main thing I'm pointing out in this line here is a very large fraction of a cell's energy budget is protein. It's not DNA, it's not RNA, it's generally protein. And this is important in ciliates, but ciliates have this giant thing called the macronucleus. All the chromosomes are replicated about 1,000 times, and yet still the cost of the macronucleus is pretty small compared to all the proteins inside the cell. Membranes, again, are pretty expensive. In your average ciliate cell, all the lipid membranes is about 10 to 20% of your budget. It declines with increasing cell size because the surface area volume ratio goes down. And then you can even think of certain activities inside cells. Osmoregulation is a big problem for cells living in an aquatic situation. And it becomes a bigger problem as the cells get larger and larger. There's more membrane for water to be leaking through. And for large ciliates, up to 40% of your energy budget can be spent on osmoregulation. They have these contractile vacuoles that are just, water's constantly coming in and it's constantly being pumped out by the contractile vacuole, which takes energy. And then finally, there's the cost of motility. And I'll just end with a slide on the cost of motility in a bunch of different organisms, showing you another one of these universal scaling relationships. A lot of organisms, single-celled organisms, can swim, and that's true of both bacteria and eukaryotes. They swim using flagella, or we sometimes use the word cilia in eukaryotes. And what you may not know is that the flagellum evolved de novo independently in bacteria and in eukaryotes. We have totally different, we both use flagella, but they have totally different mechanisms of operation. So in bacteria, there may be multiple flagella on a cell, and they move in this rotary propeller-like fashion. They do that because they are connected to a rotary motor that operates something like the proton motive force, not unlike the situation with ATP synthase. Totally different molecules are used in here than in the eukaryotic flagellum. The eukaryotic flagellum is composed of tubulins and motor proteins that slide up and down, and eukaryotic flagella move in more of a whip-like fashion. So very, very different structural basis to the swimming apparatus, and a very, very different mechanism of moving the flagella in these two cases. And yet, if you look at the measure of the efficiency of swimming in bacteria, heterotrophic flagellates, these are all eukaryotes, typically have just 2 flagella, and then ciliates, which are coated with flagella all over the cell surface area. We have this universal scaling relationship. And the measure here on the y-axis, this is the cost of building all the flagella in a cell. Oh, let me back up for a second. What we're measuring is the swimming speed in cell lengths per ATP it takes to construct the swimming apparatus. Okay, so that's the swimming, you're thinking about swimming speed per unit length, which seems like the ecological value that would be most important to an organism, and per unit ATP investment. And as you move to the right, you can see that this declines. It's almost inverse relationship, but everybody falls on the same line, whether you're a bacterium or eukaryote. So again, it's another one of these universal scaling relationships, even though the underlying swimming motility mechanism is completely different. So a lot of unanswered questions here in how we extend cellular bioenergetics to evolutionary cell biology, but I think it's at the heart of a lot of issues if we're trying to understand, on the one hand, diversification of morphologies and physiologies of cells, but on the other hand, trying to understand these universal scaling relationships that transcend species boundaries. I think that takes me to the end. So
Thank
I'll
you very
stop
much.
sharing. If there's any questions.
Yeah, we have time for a few questions. Over there, I don't know if somebody can pass your microphone.
I have a very general question pertaining to costs. To the extent that you are trying to use these arguments for a theory of the evolution of complexity, shouldn't you be looking at marginal costs rather than absolute costs, which you've been, I think, doing all along? And I say that for 2 reasons. One is you have interactions between each of these elements that you talked about, lipids or proteins or even DNA molecules. So the marginal cost really will depend a lot on who is talking to whom. And secondly, of course, the marginal cost comes in because once you have evolved a certain level of complexity, it may be cheaper to keep going in the same direction, so to speak, rather than to go back and reduce the absolute cost of doing something else.
Well, there's a couple parts to this. I guess I'm not quite clear on what you mean by marginal costs.
Well, what I mean by marginal cost is the extra cost associated with doing something, let's say adding a tryptophan in place of a glycine in a protein, when you already have a certain number of glycines and the protein is doing something.
Well, I'm not trying to get at the functional significance of the amino acid changes, if I'm interpreting you correctly.
No,
and then there's the issue.
Sorry, neither am I. All I'm asking is, when you count the cost of replacing a certain number of glycines by a certain number of tryptophans, can you simply add up the total number and multiply by the unit cost? Because the costs may be quite different depending on where you are.
Well, if there's multiple exchanges, then yeah, the cost would be greater, or the advantage would be greater switching from tryptophan to glycine. But in general, we don't expect evolution to operate like that. It's usually operating at a scale where there's one amino acid substitution at a time, and then there's a long waiting time for the next one. So they're not going to be compounded on top of each other. And then the complexity issue that you raise, this gets into a very large can of worms. I in general, and I think there's a lot of evidence for it. I mean, we tend to think that more complex organisms are in some way better off, but there's really no evidence of that whatsoever. In fact, if you make an organism more complex, there's generally more ways to break it than you had before. And it's interesting that organisms like bacteria, which where natural selection is most efficient, they don't go down the complexity route very, very far.
Other questions? Try to get a microphone.
Curious about your latest, say, power law relation between the speed and the cell volume. Can we speculate about the fact that essentially you have a certain nutrient gradient that is valid for every organism? So essentially the larger cells have somehow less distance to do in order to hunt for nutrients with respect to smaller ones, and this is why the speed evolved differently.
Yeah, it's a good question. That was one, I think the reason we divide by cell length is one reason we divided by cell length for the very reason I think you're talking about. It puts it more on the scale of the organism's biology, right? The length of the cell relative to the absolute distance. But these are maximum swimming speeds too. This is as fast as you can go. And I guess one thing I failed to mention here is how inefficient swimming turns out to be. You can measure the in principle, you can measure the cost of motility using some physics, Stokes' law and so on. You can think about how much energy does it take to drag a cell through the water. And then you can calculate how much it actually costs to swim. And the efficiency tends to be on the order of 1%. In one case, it's been as low as 0.1% efficiency. So swimming is really quite remarkably inefficient. We don't really understand, why that is. But the argument here for this gradient is as you move to the right, you're not just getting bigger, but natural selection is getting less efficient. And that's one reason we think that you're going downhill. It could be that for this all to fit, you would have to have some kind of universal scaling relationship, I guess, with respect to the ecological setting that all these things have that would pertain to everybody.
Yeah, Vijay.
Mike, you mentioned the costs of making molecules of various kinds, but I wonder there's also another cost of putting the molecules in particular configurations. I mean, a random spatial distribution of molecules is not a functional use for a cell. I guess there is cost involved in not just making the molecules, but also assembling them in functionally useful. How does that compare with the cost of making them, maintaining them? Perhaps the cost of running the functional units is much more than the cost of making the molecules themselves.
Well, I gave you the cost of making the building blocks, but if you want to make proteins, so average amino acid costs about 30 ATPs. And if you want to string them together to assemble them into a protein, you have to expend about 4 more. So maybe, roughly 10% of the biosynthetic cost of the building blocks, roughly 10% of that is the assembly cost. That's for a single polypeptide chain. Most proteins don't operate as monomers, but they assemble as dimers or trimers or tetramers. I don't think there's very much energy involved in that. There's no direct energy expenditure in that. It's just a matter of encountering each other. But if you want to be a multimer, you have to make more monomers just to increase the encounter rate of the monomeric subunits in cells. So that's an additional cost.
No, I was thinking of even macromolecular structures, let's say the cytoskeleton, where there's huge polymeric structures there. And many of them do require ATP or GTP to assemble.
To assemble and yeah, disassemble. So there's a treadmilling thing and so on. So yeah, there's, in fact, one of my postdocs is working on this, is how much energy is expended in the cytoskeleton in different contexts. And for tubulins, of course, that went into our estimates of the cost of flagella in eukaryotes.
Okay, thanks.
So from the drift barrier slide, as the organism goes from simpler to complex forms, the drift barrier makes a shift from left to right, in a general observation. So can we say that simpler organisms or prokaryotes are more likely to evolutionary changes than the complex ones? Can we infer it from that graph?
Well, bacteria and really small eukaryotes can take advantage of mutations of small effect, much more than us big clunky vertebrates and land plants. Selection is much less efficient. I would say that our evolutionary changes have to be much more coarse-grained because the mutational effects have to be large enough that they can be perceived by natural selection. So I think in bacteria, bacteria that have effective sizes around 10 to the 9th can probably feel almost every mutation that occurs. There must be almost nothing that's what we call effectively neutral. So many kinds of mutations they can take advantage of that we can't.
Thank you, sir.
So going back to this relationship between velocity and energy expenditure while swimming. So basically, I wonder, right, if so the y-axis was scaled by cell length. So I wonder, that means I think this could be explained if you assume, right, a rod-shaped, kind of roughly rod-shaped cell, then you have kind of an area at the front, right, that defines the resistance. And then if you assume things are small, then basically your velocity is probably proportional to the force because inertia doesn't really play a role. And I think then, right, you would get probably this relationship from very basic physics, because your volume, like if you rescale the axis, you could plot it as area versus energy, and then.
Yeah, one thing I might not have made clear is so this is swimming speed per energetic investment, and that's in the swimming apparatus itself. That's scaled to the cost of making all the flagella.
Okay, so
it's not just the energetic cost.
I see.
It's not just the cost of running the machinery. It's the cost of building the machinery.
I see, thanks.
So if there are no more questions, maybe I have one last comment. So I feel like these estimates are quite useful. Can you point us to a review or a general reference when we can find these numbers?
Yeah, they'll be in those book chapters that I put on a slide yesterday.
Okay. So we can look at, okay. Thanks.
Yeah, you're all welcome to work through the cost. There's some sections called foundation sections. They're sort of like appendices. That was a lot of work.
Can imagine.
No guarantee that we haven't made mistakes, but we tried to lay out the recipes and the step-by-step procedures, and so that someone could retrace our steps and correct us if we happen to be wrong. I don't think we're hugely off, but there's a lot of nuances involved.
Okay, thanks a lot. So if there are no more questions, let's thank Mike for the very nice lecture.